So I follow up on Ariel's presentation, and you will see also a lot of examples about some peak splitting and phospho consideration. But in general, you can use this kind of as a tutorial how to build high-quality spectral libraries. So I made this presentation specifically oriented towards SWAS, but this would be the exact same consideration you might want to think about if you want to do a very good quality assay library for SRM. And you can even apply this in principle to do spectra searching if you use a spectral library searching for shotgun data. So most of these considerations are anyway important for any type of spectral library matching that you would do either for shotgun, SRM, or SWAS. As soon as you want to have accurate retention time information, relative intensity, etc., this is, I think, a very important consideration. So I want to comment that the assay library is a very central part of our workflow, because once you have acquired the data, so I showed you this morning that the acquisition of the data itself is not very complicated. The machine acquires basically everything, all the mass range across all the retention time. But what's very critical to get a very good extraction, a very good quality of data analysis is also to have a very good quality of assay library. Because this is going to drive what you're going to extract and what you're going to query inside your very complex MSMS spectra datasets. So when I speak about spectral library, I think you've seen this already a couple of times in the context of SRM. So this would be basically a assay library for SRM. You have a transition list, which are basically for each peptide, the mass of the precursor of that peptide. This is the Q1 mass. Then you have the Q3 mass, which is the mass of these specific fragment ions. So these are the transitions that you would measure in SRM. So Y5, 7, 8, 10. In SRM, you also need to specify for the machines the collision energy and the dwell time for which you want to spend on each transition. And these are mostly relevant information for SM acquisition. And a very important consideration you might remember is that whether you do SRM targeted acquisition or SWAS targeted extraction, your peptides can only be tested and queried only if they are included in your assay library. So in SRM, of course, you need to have that because that's what you're going to acquire. But in SWAS, you also need to have this kind of assay list in order to query also your peptides. And only the peptide that you have in your library, you will be able to detect or to test if they are present in your sample. So in SWAS, you actually don't care so much at this level about the collision energy and dwell time, but you need a couple of other information that are very important for the extractions. And these informations are basically the precursor charge, the fragment charge, the relative fragment intensity, retention time. And they have different purposes. So for example, like the relative intensities and fragments, these are very relevant information for you to test. What are the most important fragments to test your peptides? So you want only to acquire or to query for the most intense peptides. So you might take the most intense fragments. So you might take the top 4 most intense fragments of that peptide, which are these relative fragment intensities. You want to test them with the right charge state in order to query them in your data. You also want to query them at the right retention time. So this will allow you basically to decrease a little bit your window of retention time. So in SRM, you might want this information also in order to do some kind of scheduling in SRM acquisition. Or in SWAS, you might want to restrict your window to a 5 or 10 minutes windows in order to gain in some specificity instead of extracting the whole entire chromatogram. And again, the relative fragment in ion intensities are very important information when you do some kind of peak identification. So you've seen in the talk of Ariel that the relative fragment ion intensities allows you to differentiate between the different phosphoisoforms that you have. And for any kind of peptides, the ordering of these fragment ion intensities, so that the Y5 at this mass is the highest intense one, and the ratio between those relative intensities are very important information when you want to be sure that you have the right peak detected. So all this is basically, as I said, it's the idea is that once you build this assay library, you want your assay library to look as similar as possible to what you're going to look for in your targeted acquisition or in your targeted extraction. So this relative fragment intensity, the retention times, you want to have them as accurate and as close as possible to what's going to be the one that you're going to look in your targeted proteomic assay. And this is all the trick of getting this information as close as possible to your final measurement that you're going to do. So I'm just going to present you just very quickly the workflow. So this is nothing really complicated by itself, but it's a lot of little tips and tricks that you need to know a little bit. So when you start, in general, to make a library, you would typically do a sample preparation, a kind of a shotgun analysis. So sometimes the peptide will be picked once or twice. You acquire the MSMS spectra for those peptides. You get the shotgun identification by regular target decoy database search in shotgun. So you get the same peptide identified once in run 1, twice in run 2. You do some kind of a consensus merging, so you want to get the best quality you can. I'm going to speak about that as well. So you want to get basically an average picture, the best you can get. So you average these 3 pictures into one consensus picture. And then from this consensus picture, you might want to consider the 6 most intense transitions in order to get this table. And all the work is basically how to get to this table with all these different steps and to get the table as close as possible to what you want to see in your SWAS extraction or in your SRM acquisition. So you want that the top ion here, Y10, is the same one that you expect from your shotgun and is the same one that you expect from your library. So you want this to be, again, this table has to look as similar as you want to the final results that you will get from the targeted proteomics. And I'm going to explain you that you have basically, so this is a relatively easy to follow workflows, but at each of these steps, so sample preparation, shotgun acquisition, consensus generation, when you generate this assay list, you have each time a little bit of little tips and tricks I want to show you today to get you, to tell you how you can get best out of your data, basically, to make this table, again, look as similar as possible to your targeted proteomics assays that you will measure. So this is a manuscript that we have been writing with Olga and Ben in our lab, and this is now still in revisions. Hopefully it will be accepted soon, and this is kind of a very long workflow with a lot of bioinformatic tools. So I'm not going to speak in details about the bioinformatic tools that we are using. These are very technical, but I want to give you at least the little tips and tricks you need at every step in order to get the best out of your data. So these tools can be adapted. Most of them you can also do, most of those things you can do in Skyline, most of those things you can do maybe with your own pipeline, being BiblioSpec. We are using Spectras because in our lab we are TPP-centric. But you can do many of these steps. You can basically apply it to any of the tools you might already be using. You just need to tweak and think about these different tips a little bit in your own workflow. So I will try to walk you through these different steps. And at each step, my aim for today is to give you a couple of tips and tricks just to think about. And when you will build your own libraries, you might use this information as a repository to try to tweak your system to get as good quality assays as possible. So the first step is about the sample prep. So the sample prep is, again, the aim is that you want your peptide to be in your assay list, and of course, because we're doing targeted proteomics, only peptides that are in our assay library, you will be able to detect them in targeted proteomics. And you want to generate extensive peptide assays, as extensive as possible, because you might want to query as many peptides as possible. So first of all, if you do, for example, like think about your experiment, you do a time course or an inhibition of a certain enzymes or kinases or whatever, and you know that the proteins that you will quantify in time point 1 will be different than proteins that you will quantify in time point after 1 hour, after 24 hours. So you have to think that you might have proteins increasing, you might have protein decreasing, And if you want to acquire a library of as many peptides and proteins as possible, which are representative of your entire biological space, you also want to sample all possible biological conditions that you want to measure as well. So if you have a protein which is only expressed in late time points, you also need to make a library of the first and of the late time points, etc. So you want that your samples for which you make the library on will cover the entire biological space that you want to quantify by targeted proteomics. So if you're missing the late time points in your shotgun library, you're also likely to miss the proteins which are expressed there. So when you acquire your library, acquire them in as many representative biological samples that you have for your own experiments. In addition to that, you might also want to fractionate your samples when you generate your library in order to make as extensive libraries as possible. And one fractionation that we have used extensively in our lab, it's just an example, it's the object electrophoresis, where you basically fractionate the peptides by isoelectric focusing, by isoelectric points. And there, basically, instead of having one sample that you inject in shotgun, you basically fractionate it to 12 fractions, for example. So each sample can be injected 10 times or 12 times more concentrated, and therefore you gain in sensitivity in the shotgun library, and therefore you also gain in acquiring more assays that you would do if you would just shoot one single time points in shotgun. So fractionation, basically, it helps the deeper sequencing of your sample. Another possibility that we have used extensively in our lab is once you reach completion of all the proteins you can identify in your samples, you might also want to complement these assays also with peptides for which you haven't managed to identify them endogenously in your samples. So you might want interested in proteins which you did not manage to detect by shotgun even after fractionation, and for those you might want to order the synthetic peptides and then measure the assays. So this has been covered by other people already. So this will help you to complement your library with additional assays. And then the final point is you might also want to complement this with completely theoretical fragmentation predictors. So this is basically going to be the least reproducible, let's say, the least faithful representation of your peptide fragmentation, because these are usually relying on algorithms to predict the fragment ions, and this might be the worst, the furthest away from the reality when you measure your samples. But basically with these pipelines, acquire all the biological conditions you can, fractionate, complement with synthetic peptides, you basically already cover as much as possible your space of proteins you want to quantify. The second point that I think you had a special talk about this is about the retention times. So in order to share or even extend for yourself later your libraries, you want to have also the retention time included inside your library generation. And for this, we use basically spiked in reference time peptides. You heard about the IRT peptides normalization, and this we use basically for retention time normalization and for retention time splitting. And the idea is that, for example, if you acquire in the first condition here with a certain gradient of 30 minutes, your peptide is coming between retention time peptides 9 and 10, 8 and 9, for example, at certain times. You want to use this retention time information in a different gradient for targeted proteomics, where this is going to be like a 40 minutes or 2 hours gradient. And therefore, for this, you basically rely on these red retention time peptides to realign your gradient lengths, to realign exactly where you expect your peptide retention times. So the peptide retention time is projecting in this unitless space, which is this IRT space. So this 21 minutes is basically translated to 62 IRT values, and these 62 IRT values then translates to another retention time of 29 minutes in the other conditions. So this allows you basically to transfer assays acquired in a certain conditions in shotgun library generation to basically targeted proteomics in a completely different gradient conditions. So these are very important if you want really to extend the information of your libraries and to use this like not limited on the same gradient and on the same lab, but basically to transfer your assays across different conditions. So the second step I'm going to speak now about is about the shotgun acquisition. So the idea of the shotgun acquisition is you want to have your machine in shotgun performing as close as possible to your data independent or to your SRM fragmentation conditions. And the idea again is that you want to mimic with shotgun, you want to mimic as close as possible the fragmentation that you're going to get in targeted proteomics. So for this, we recommend really strongly to use, if you can, the same instrument for library generation and for targeted proteomics. So sometimes if you're lucky, if you have, for example, I don't know, like a Q-trap where you can basically build a library and use it as a triple quadruple to do SRM, it's better to do it on the same machines because you're sure, because of the collision cell geometry, you get as close possible fragmentation patterns as you get in your shotgun library and as you get in your targeted proteomics measurements. We also show that some instruments can be transferable between some each other. So for example, if you do shotgun acquisition on an orbit trap, on a Q-exactive with HCD mode or an orbit trap with HCD mode, you're still very comparable to a quadruple fragmentation on the different type of instruments. So for this, we have published another paper where we basically tested and assessed the transferability, the reproducibility of the different fragmentations between instruments. And we showed that in this case, for example, we compare it to SWAS acquired on the triple TOF, and you see that these basically violin plots represent the distributions of similarity. And you get the score is as high as possible to 1, which means that the shotgun is as close as possible in terms of fragmentation reproducibility to the SWAS fragmentation. And you see here the triple TOF shotgun is the closest and the highest scoring towards the triple TOF in SWAS mode, targeted proteomics. And the second, actually very close, is actually the orbit trap in HCD, is also very similar. But as soon as you start to introduce like CID collisions instead of beam type fragmentations, you start basically to lose a lot the fragmentation similarity between the machines, and therefore your assay will look in shotgun much more different than the assay that you would acquire in quadruples or in Q-exact. Yeah.
Did you ever do a comparison of actual SWAT peaks and SWAT peaks?
Yes, I might have it in the supplementary. Okay. So this is very important point. So this is what we call the portability, and we have the intrinsic reproducibility. So this is also in the same paper, and this is actually much better than the shotgun reproducibility. And I think this is due to the fact that in SWAS, or in targeted proteomics, you integrate the area, and you're much more accurate by doing this than by just comparing the relative intensity of a single snapshot, which you get by shotgun. So the SWAS is the most reproducible intrinsically, and the second most reproducible is actually the ORB in HCD. So it's actually more reproducible than the triple TOF itself. But still, even though the triple TOF in itself is not so reproducible in shotgun, it's still the most closest to what you get in SWAS. So this is all, when I try to describe this in this paper, it's very technical, it's very complicated to describe maybe, but actually the thermo machine are the best reproducible in shotgun, but they are a little bit further away to SWAS than the triple TOF itself.
So I guess then my follow-up question is, why would you stop with Spectra? Why wouldn't you use Spectra to seed swath measurements that you then use as your assay library?
Yeah, so we started to think about this. So in a second iteration, you would be actually even more closer to the reality. You're absolutely right. But in the first step, in order to detect the peak, you still need to do some kind of a shotgun test. I mean, the other possibility is to do what I said this morning, to do some kind of a MSC kind of identification of your SWAS data, and then to use this as identification and as assay generation for the SWAS extraction. So they would be actually the best type of scenario in order to get the closest assay from the beginning on from the same instrument, from the exact same file actually. So you get as close as possible you get. But if you're interested, so we try to describe this in this paper. So this is comparing the intrinsic reproducibility of the machine in terms of fragmentation. So how reproducible is my machine for fragmentation? How transferable is my assay from one instrument to another instrument? And it was a master student in our group that developed this. It's a tool. It's running in Python, where you can basically provide libraries and you can generate these plots very simply. And then you can compare library between each other to see how reproducible you are, how transferable you are between instruments. So you can have a look if you are interested. This was validated basically on fragmentation level, and it was also validated by colleagues in Seattle. And you see actually also the influence of the fragmentation parameters that you use in your library creation. So you heard about the parameters important for SRM acquisition, and these are parameters that you might want to use for shotgun acquisition in order to get the best possible spectra library, again, for targeted proteomics. And what we recommend is actually not to try to operate the machine as fast as possible, but rather to be a little bit more conservative, use a little bit longer accumulation time. So we recommend like 150 milliseconds, because actually you see that by increasing the accumulation time for MSMS spectra, you actually gain in confidence, you gain in reproducibility, and you gain in transferability of your assays to your targeted proteomics. And also we recommend to acquire more spectra, so more replicates, etc. And this we can also do by reducing the dynamic exclusion of your shotgun to get to about half of the peak width. So this is most important for QTOFs. Maybe for Q exactives, it's also, as soon as you have an ion trap that can collect the ions, it's not so dramatic. But for quadruple tap instruments, you might also want to have your peak selected at the beginning of the peak, and also at the top of the peak, where you would get the best possible fragmentation spectra possible for your peptide. And as you can see here, it directly correlates with the number of identification and the quality of the MSMS spectra that you get. So more injections, more spectra, more accumulation time, all this will increase the quality of your MSMS spectra, fragmentation, and this will directly translate into better assays for your targeted proteomics. And the last very important point, actually, that was overlooked in some studies is that the collision energy is also very important. So you know in SRM that you also optimize very much your collision energy, your declustering potential, to have the best you can out of your fragments. And here, when you build your library in Shotgun, you also want to be as close as possible in terms of fragmentation to your DIA or to your targeted proteomic experiment. And therefore, we actually want to use the as close as possible collision energy equations. So for example, here in SWAS, we use basically rolling collision energy. So this is designed by this equation. So this is the slope multiplied by the m over z of the precursor plus an offset. And basically, in general, if you open your instrument, so this is from the triple top, this is from the Q exactive. Sometimes some instruments, for example, allow you different equations for different charge states. And because in SWAS, or because in being on the triple top on the Q exactive, you co-select and co-fragment many precursors simultaneously, you cannot change the collision energy of your window based on the precursor that is going to be inside. So if it's a doubly or triply charged or quadruply charged inside your window, you're going to apply the same collision energy in DIA. And therefore, you want actually the shotgun also to have the same collision energy regardless of the charge state, and that the collision energy scheme for the fragmentation in shotgun should also be the exact same scheme that you use in your SWAS or DIA acquisition methods. So this directly influences the fragmentation patterns that you get in shotgun, and this is allowing you to get basically the closest possible fragmentation patterns to your targeted proteomic experiments. So you can do this on the triple top, you can do this on the Q exactive, and you have to edit these collision parameters, ramping, etc., to be the same in shotgun and the same in your DIA acquisition modes. And therefore, you will get basically the most similar fragmentation patterns between both modes. So this was for shotgun acquisition. So now you have acquired your data, you have your raw data, and what most search engines need at this step, they don't manage to work really directly with the raw data, which is profiled from your mass spec instruments. The first step you might not be aware of, maybe it's done automatically by your pipeline that you use in your lab. It's to convert this raw profile files into centroid picklist format that is going to be searched by the search engines. And to our surprise, actually, we figured out that the converters might actually work or perform very differently depending on the type of integration that you do for the peak picking. And during this centroiding, for example, some converters use the peak apex as in order to convert the row profiles in order to centroid them. So sometimes they use the peak apex of the profile, sometimes they use the peak area of the profiles, sometimes they integrate more noise or less noise, remove some backgrounds. And this actually affects very dramatically the performance of your spectra. And the first thing I want to comment about, this is also something that we checked in the past, that you have actually converters that also do de-isotoping. So in principle, this in Shotgun you do if you want to help the search engines, because some search engines are pretty bad in identifying like doubly charged Y ions or doubly charged B ions. So some converters, they do something smart. They say, I recognize this is a doubly charged fragment, so I'm going to multiply this mass by 2, and I'm going to put the mass somewhere else. The problem that once you do these libraries, you get something like this. So you get a peak, you have your 4 most intense positions for your peptide, you see 3 very well colluting, and then there is a 4th one which is completely flat. When I started to look into that, I got very puzzled because I was wondering what was going on. I opened my SWAS MSMS Spectra. So in my library, I was expecting the very strong, actually was the most intense fragment here for the Y12, and there was clearly no fragment at all in this Y12 region. So then I looked into my Shotgun data, and my Shotgun data search results had a very intense peak at Y12. Then I got very confused, and I said, why is it in my Shotgun and why it's not anymore in my SWAS? And then actually I went back to this exact raw spectra. This was the centroiding results, and this was the profile original spectra. And as you can see here, the Y12 is absent. So what the software did, it figured out that this was my Y12 doubly charged. It thought, okay, I'm smart, I'm going to do deconvolutions. So this is now not anymore Y12 doubly charged, it's Y12 singly charged. And this you carry because you take your top 4 to your assay libraries. And here, this is what you carry over to your assay library. But here, there is no fragment. It's just an artifact of deconvolution. And therefore, I commented this was done on the ABSIX converters. And I told this to the ABSIX people. They claim it's a feature from the converter. I would say it's a feature, but you don't want to use it for assay library generation. Might be good for shotgun searching. It's fine. But as soon as you want to do something else, like assay generation, you want to use real fragments. You don't want to use like deconvoluted de-isotope pictures, right? So be careful, and once you know a little bit how your converter behaves, you have to think a bit what is it doing and what you want to avoid, because you want to do something special about the data extraction. So this is the first step about the de-isotoping. The second step is that we figured out that you get better results if you use centroiding algorithms that use peak areas. So this is to show you an example. So I want to point out, so this might not look very clear from the beginning. These 3 pictures are the same raw MSMS spectra. So this is the same scan number, raw data processed with 3 different converters. And as you can see, the pictures looks actually pretty different. So this one here, the top ion is the Y10. Here the top ion is the Y6. And you will not even guess by looking at the spectra that this is the same species. It's the same raw spectra, the same profile spectra, just processed with different converters. So depending how the converter works, if it includes more noise, less noise, peaks by peak area, peaks by peak intensities, you get basically a completely different relative intensity for the fragments. And of course, as I told you, the aim, you want to get as close as possible to the targeted proteomics experiments, because this is what you want to target for. And therefore, you want to use the converter that gives you Y10 as the most intense fragments, because that's the one that is actually the one that you are going to target at the end. So even the same raw shotgun spectra processed by different converters will change quite drastically sometimes the relative intensity. So I took here, it's kind of an extreme case, but it happens, and therefore you need to assess. So you can also use the tool I showed you before, try different converters and see which one is actually getting you closer to the MSMA spectra from your targeted proteomic experiment. So all these are little details, but if you sum them up together, it starts to make quite of a big difference in terms of quality when you build your library. And this is an example to show you that this spectra was actually very bad in the converter here. And it's kind of getting a little bit better if you do some kind of consensus, so averaging of many, many spectra across many different measurements. But it's not, I'm going to speak about the consensus generation later, but it's not yet quite as close as, for example, other converters. So you have to be a bit, think about your pipeline, because if you want to use this shotgun in order to build your assay libraries for targeted proteomics, think about really which converters and how to do these things right. So as I showed you, you can use basically this also tool to see which converters basically performs better than some others. And this is also a nice way in order to assess the performance of your converters. And I would suggest that this is very instrument specific, and you might want actually to check this type of performance for your converters and for your pipeline for your own specific setup, for your own specific instruments in your lab. So this was for the data conversions. Now I'm going to speak about data searching. So this is basically simple parameters that everybody uses in Shotgun. So we recommend to use a database containing like a peptide sequence with the retention time peptides in order to be searched. These IRT peptides, in order to realign your runs, should be embedded in your database. This allows you to automatically detect them on the fly and to perform a realignment very easily. We use also decoys database by pseudo-reversing. This was commented by other paper that this is most sensitive way to do database searching in Shotgun. We use sumitripsin as a search option, which gives a little bit better confidence, even though you might in SRM want not to use sumitriptic peptides. We still think that they are very sometimes biologically relevant if you think about it. And one thing that we also started to do a lot in our lab is basically to combine the results of multiple search engines. So you know this type of plots from many different studies. This was an example where you have basically different search engines, and actually the overlap is only 6,000, and each of the search engine identifies 6,000 additional ones that none of the other ones identified. So you know that the reality is not the intersection. The reality is also not the union of those, because then you would be also too loose. But you need some tools, like for example, there is this iProphet that was developed also in Rudy's group. And this basically managed to compute certain kind of confidence when you combine multiple search engines together. And this is the way basically we consolidate our results. And we basically get something between the intersection and the union. So we get something like about this range that contains the intersection and most of the ions of the other ones, etc. So we use kind of a multiple search strategy in order to, again, get as many peptides as possible when we generate your libraries. And the last step is actually to use a very stringent score cutoff. When you build your libraries, especially when you build huge libraries, you might want to control the FDR at this level very, very closely. So for this, we use also a tool that was developed in our labs called Mayu, which is not the PSM FDR. When you think like I want to have my data at 1% for discovery rate, we actually use 1% protein for discovery rate. And therefore, we are a little bit more stringent than the usual criteria for people that just do shotgun protein IDs. And the reason for that is that there are actually all mistakes that you have in your shotgun library, you will carry them out in your targeted extraction. So this is an example where you have a wrong peptide assigned to your sequence. So for example, I mean, the most easy case, for example, would be a decoy peptide sequence. But if your decoy peptide sequence turns out that it's a real peptide, you will actually validate the false peptide identification in your swath, because even though it's a wrong peptide, the fragments still belong together. It was just a wrong ID because the database matching was wrong, or because maybe the peptide was not in your database. So you got a wrong labeling. So the sequence of your peptides assigned to the spectra was wrong, but the spectra was indeed coming from the right precursors, and the top 6 fragments should belong to the same precursors. And in swath, because swath doesn't give you the way to check whether the identification is correct, it just tells you is this assay present in my sample? And I say, yes, this assay is present in my sample. Whether the peptide sequence is correct or not, SWAS won't tell you. So targeted proteomics just validates the existence of your assays. And in order to control for this, we want to be as stringent as possible in the peptide FDR estimations. The other type of mistakes that you carry from shotgun to SWAS are like multiplex spectra, which basically are composite spectra of two different precursors that co-fragments together. And because they co-fragments together, the search engine was confused and then assigned the wrong sequence. And these ones we can actually manage to resolve them by SWAS, because if you extract the top 6, you actually realize that the top 6 don't collude together. You have 3 fragments that elute on the left, 3 fragments that elute on the right, and then you realize that this was actually a composite spectra that got the wrong sequence identified. So these ones we can manage to solve them by SWAS because we actually realized that the identification from shotgun was wrong, but this type of false database results, but from the still a right precursor identification, from the right precursor fragmentation, but wrong identification, these ones we cannot solve them in SWAS. And therefore, we have to be very stringent in order not to pass too many mistakes here in your SWAS extractions. And this we do with Mayu. And what Mayu does, it's a very simple principle, is that the more spectra you acquire, you're more likely you're going to your match to spread over false protein identifications. And this is a very simple cartoon to show you that if you have each of these round dots are basically protein IDs, and you see that if you shoot your sample once in shotgun, you will basically have the true protein, the true spectra that always kind of cumulates at the same protein IDs, but the false false matches, the false spectra identifications, basically they are random matches. So some of them accumulate on top of true protein IDs, but most of them actually accumulate randomly across all the rest of your protein database. So you will have in principle as many false ID in your protein here than in your decoy matches. So you can still control this a little bit, but the problem is that as soon as you start to accumulate and acquire many MSMS spectra in shotgun, like fractionations or multiple injections of the same sample, you actually continue adding and accumulating true positives for the same proteins, which are really present. So this one gets more and more validated. But you see here the random matches, which are bad spectra, they will continue basically to spread over the protein identifications. So the false protein identifications will basically get more and more random hits. And at the end, you will basically get 100% apparent coverage of protein identifications, while actually most of them are wrong. They're just random matches. And the true matches only actually cluster in the right protein ID. So this is a big problem when people do like huge shotgun experiments or huge type of database search engines, that if you don't control very well for protein false discovery rate, you might think I'm still right with the peptide and PSM, peptide spectrum matches false discovery rate, but your protein false discovery rate is already exploding because you're actually accumulating too many and spreading over too many false IDs across your proteins. And what Mayu does is basically controlling this by some statistical methods, and you basically switch the idea from false discovery rate on the spectra matches to the false discovery rate on the protein matches to be much more conservative in the assays that you transfer to your library. And yeah.
Do you go back at all to the library and do that again? Because
in principle, as soon as you add more shotgun,
you have the same problem with adding to a library.
Yeah.
The false discoveries are going to accumulate faster than the
I think if you want to increase your library size, you anyway need to, so here I'm also going to speak about like the library generation, the consensus generation, the IRT merging and IRT splitting. So you need anyway probably to go through the entire pipeline. So each time you want to extend your library, you might need to reprocess kind of everything, I guess. That's the best way. But this is quite critical because, for example, yeah, I mean, when they started to do like the kind of pan-human, like huge database searching, if you don't control this very carefully, you might think, wow, I get a lot of proteins IDs, but actually maybe most of them might be random hits, right? So you have to be a little bit careful. And when we build libraries for targeted proteomics, you want to be as conservative as possible. The next step is basically the spectral library generation. And at this step, again, the idea is that you want to compile your spectral library to look, again, as similar as possible. So you want to generate, first of all, you compile all your spectra into what we call the redundant libraries, which are above your protein FDR cutoff. And then you want to transform all your spectra retention times to a normalized retention time space. So at this step, we basically transform everything to IRT space. And here, I'm going to comment about the splitting of the retention times. So this is something which I implemented originally for phospho datasets mostly, but actually you will see some examples of normal peptides that also split it. So this is a little bit the same concept that Ariel presented earlier today. So here you would have like a good case scenario where you have one peak and the same protein, the same peptide identifications. So since it's the same peptide, you actually get very similar spectra. So the spectra peaked at retention time 1 and spectra peaked at retention time 2 are very, very similar. It's the same species. The retention time also within the peak width, so they're very similar. So when you build your consensus here, you basically average your retention time and you average your fragmentation spectra. So you get from this one assay with average retention time and average relative intensity of your fragments. But you're good because it's the same species, so you extract one assay in your targeted proteomics experiment. Problem arises when you have actually two peaks, so this is what I had showed already, and both peaks actually got the same protein peptide identifications. So the problem is that these are obviously two different species, and the problem that because they have the same peptide sequence and very different fragmentation and very different retention times, when you build your consensus library, the problem is that the software cannot know that these are two different species. So what the software does is that it's finding it's the same peptide sequence. I should merge those. So I'm averaging the retention time exactly like here. I'm averaging the relative intensities exactly like here. But by doing this, you realize that your retention time will be wrong because the retention time will be in the middle of both peaks. The relative intensity will be wrong because the average intensity will be completely screwing up both individual spectra. So you get an assay with wrong retention time, with wrong relative intensities. And this is what you're going to try to extract in your targeted proteomics assay. And this you don't want to do because you have wrong retention time and wrong relative intensities. So in order to avoid that, which is quite dramatic when we do phosphopeptide analysis, so I developed a pipeline where we basically, if you figured out that your spectra are too far away in retention times, you realize that they should not be merged together. They are beyond my peak widths that I expect in my chromatography. And then you tell the softwares, I don't want you to merge them, I want you to split them away. So this is an example of a long phosphopeptides here. You see the first species was identified at 45 minutes, second one at 50 minutes, so they're 5 minutes apart. And then I thought, I know my peak width should not be long 30 seconds, so it's not possible that the same peptide IDs is identified 5 minutes apart from each other. So at this stage, basically, instead of doing a simple merging and consensus generation like you would do in normal libraries, what I do here, I basically split both species apart. So I say this species is assay 1, this species is at the same, it has the same peptide identification from the shotgun search, but I don't believe it is right, so I call it assay 2. And then I process them completely independently from each other, and then I leave assay 1 here with the correct retention time with the correct relative intensities, and I keep the assay 2 with the correct retention time and correct relative intensities, and I manage to identify both peaks. So this is something which is not very well taken into consideration yet on normal. Yeah. Yeah. Go.
5 minutes.
Huh?
So 5 minutes left.
Yeah. Okay. So this is something which is not very well taken into consideration into the current pipelines, and this is mostly affecting phosphopeptides so far. So this is something I was very careful in designing for phospho analysis. But if you do actually for naked peptides, there are actually many cases where you find peptides also like 5 minutes apart. These are naked peptide sequence. There is no obvious modifications where you would get this is actually a wrong. So in this case, I think this is some kind of strange LC pigtailing that understand that this peptide here has the same ID than this one, but it's just very far away. Maybe it's pigtailing. But sometimes the peaks are very clearly like 5 minutes apart. This is not pigtailing. This is what Ariel suggested might be in-source fragmentation. And there are even where, so in this case, the in-source fragmentation, you can be sure because you have the same relative intensity for both peaks. So this tells you that it's probably inside the machine, the same species that flies. But sometimes you actually have even weird species where you see that it's actually different fragmentation. So we actually have transitions crossing each other. So inside the collision cells, these are obviously different species. And so this is something that you see a lot now in SWAS when you actually look into your data closely. And if you're not careful about splitting these assays away, you would just basically merge those two things together, extract one assays, and just get one peak. And here, by splitting this, you start to see very weird things happening, and we still don't know what this is. And yeah.
Another thing that we do in Skyline is that I saw a number of peptides in several runs where things were coming out in the wash. And so you'd have an ID and then you'd have something way far away in the wash. So I have an algorithm to detect that.
Okay, yeah.
Throw those
so you don't
away.
want to merge them together with your right retention time.
Yeah,
but
basically if it's closer to the end than it is to the other peaks, we just
trash it,
completely
yeah.
discard.
But I still think some of them might have some biological relevance, but I haven't yet had time to investigate this in detail. But I think it's very interesting what we start to see here. And the last thing is about consolidating these multiple spectra. So we try to compare like best replicates to top 5, top 10. And I think we actually do gain by doing like a consensus rather than the best replicate. You do gain in reproducibility of your fragmentation. You do gain in identification of your peptide by targeted proteomics. So it's really worth to actually acquire more spectra and do a general consensus and averaging at this level. So this we also check with our tool. And the last step is basically how to make from the Spectra Library then the final table. So here you want to use like 6 transitions. This is what we basically want to recommend. This is high enough to ensure specificity, but not too high to introduce too much noise. And this was already a recommendation also for SRM in this paper. So 6 transitions for light. You want to use the same number of transitions for all the assays. This is very important if you do the statistical, like high throughput scoring softwares that I showed you this morning. You want to use the same number of transitions in order for the distributions to look as homogeneous as possible. You want to use B and Y ions, except for SILAC workflows. So in SILAC, especially in SWAS, the heavy and the light will very often fall in the same window, and therefore the B ions will overlap. So you don't want to use B ions in this case, you just want to use Y ions. And for SILAC, we also have designed a very specific pipeline where you actually want to acquire assays because you want to have the exact same transitions for heavy and light. You do not want to have a mixture of both. So you have to control very well how you build your library at this level. So we can discuss this offline if you're interested about. And what we have also implemented is basically you can also, what Ariel showed, the neutral losses are also sometimes very interesting to implement in your data sets. So you spoke about like phosphor neutral losses, but you can also think about water, ammonia losses, which are very common. And as you can see, they might account for like 3, 4, or like several percentage of your library. So if you have like a small peptides where actually it would be interesting to implement also the neutral losses, we check they're also very well conserved, so there is no problem to use the neutral losses as long as they're not too redundant with your peptide sequence. And the last recommendation that we have is basically to exclude the low mass transitions. So because in SWAS, this would be basically 1 or 2 or 3 amino acids, and this would be a combination might happen from many different peptides, so this would be too noisy for SWAS extractions. And we also exclude those which will fall in the selection and fragmentation window. So this you can also do in Skyline as well on the fly with the filtering options. You can also, we also recommend to use the same number of decoys for the peptides and to use them with a pseudo-reverse or scramble sequence. And with this is the final pipeline that hopefully you might be see in this paper if you're interested. So these are the complete tool sets and different steps that we have in this pipeline. And this is basically the summary of this is it's a lot of work, but the assay library needs to be prepared only once. So once you have done it, you're good. You can give it to other labs. You can give it to your colleagues. You can use it forever. Maybe it might be worth to spend some time to get the highest possibility of the assays. And the idea is always to get the library as close as possible to your targeted experiment. And our recommendation is really to use the same instruments, check for portability, check really what's the similarity of fragmentation, and really be critical about all these little steps that at the end can make a big difference in your quality of your library. And this is for you. I compiled a kind of a checklist. So this basically summarizes all the steps I described in all the slides. And if you go back to your lab, you can just check this list. Basically, if my samples originate from biological samples, yes. If my sample contain reference time peptides, yes. So you can use this really like kind of a checkbox in your lab to see whether all these boxes are checked, whether you're very confident that you can get the best you can out of your spectral library generation. So you can use this really as a very small summary of all the little tips and tricks I gave you today. And if you manage to check all the boxes, you could be pretty confident that you get the best library you can get out of your sample. Okay? Sorry, I was wrong again. Thank you very much.